Good afternoon, everyone, and thank you for joining the Melanoma Research Foundation for our first ever Facebook Live Meet the Researcher. We are so excited to have Dr. Andrew Aplin from Thomas Jefferson University join us this afternoon for our discussion. Before we get started, I would just like to let you all know why we are chatting with Dr. Andrew Aplin today. We will be honoring him in just a little over a week at our first ever virtual Philadelphia Gala. We're so excited to honor Dr. Aplin. He has been a huge supporter of the Melanoma Research Foundation for many years and very involved with our work. So we're really thrilled to honor him with the Humanitarian Award and celebrate all of his accomplishments on behalf of the melanoma community. My name is Kyleigh LiPira. I'm the CEO of the Melanoma Research Foundation, and I'm going to be facilitating our conversation with Dr. Aplin today. I would like to thank you all for joining us, and I would also like to do a special thanks to the sponsors of our gala for this year, our presenting sponsor, Castle Biosciences, and our industry startup sponsor, Merck. So with that, I would like to toss the baton over to Dr. Andrew Aplin, again from Thomas Jefferson University, to introduce himself.
Thank you, Kyleigh. My name's Andrew Aplin, and as Kyleigh mentioned, I serve multiple roles at Thomas Jefferson University in Philadelphia. Within the Cancer Center there, the Sidney Kimmel Cancer Center, I serve as the Associate Director for Basic Science. So I oversee a lot of the wet lab-based cancer research. I'm also the Kalbach-Newton Professor of Cancer Biology within the Department of Cancer Biology at Thomas Jefferson University. I'm the principal investigator of multiple NIH and foundation grants, and these are all very focused on seeking new therapeutic avenues for melanoma patients. I also participate in many programmatic efforts with other cancer centers and melanoma investigators around the country. For foundations, I serve a very important role on the Scientific Advisory Council for the Melanoma Research Foundation. And I also serve on the steering committee for the Society of Melanoma Research.
That's wonderful, Dr. Aplin. Thank you so much. And normally you and I would probably be seeing each other at ASCO in a few days. And of course, our world has gone virtual. So here we are. But I think it's really important for the melanoma community to understand what's going on in the research community. As an advocacy organization, we have received many calls from patients and caregivers who are expressing concerns, seeking information, just really wanting resources because of COVID-19. But something that we're not really hearing about as much is how this is impacting researchers and the work that you all are working on to help advance treatment options for patients and caregivers. So can you just share with us a little bit about what's been going on in your world since COVID-19 happened? And in particular, what is the research world like right now? How are they working through all this?
So to give you some background, first of all, I'm the principal investigator of a melanoma research lab. And that's quite a diverse group of people within my lab and a diverse range of expertise. So it ranges from genetic analyses through to immunologists and computational biologists. What I would say is that our research efforts have really kept going throughout COVID-19. So the wet lab activities have de-escalated. And what I mean by wet lab activities are the more traditional views of pipetting into tubes and looking down microscopes and culturing cells. But we've then compensated in different areas. And one of the areas that we've really worked on is computational biology and bioinformatics. And so part of my lab is really invested in looking into patient samples, biorepositories of patient samples. And there's really just a treasure trove of data associated with these melanoma samples that have been collected by different institutions, and the data sets that are within those samples are associated with those samples. So they could be data sets associated with mutations, expression profiles. And so what we've been spending a lot of time doing is really refining our bioinformatical or computational skills, and then using those skills to really delve deep into the data sets and look for patterns of response to, say, for immunotherapy in cutaneous melanoma, or looking for markers that associate with the spread to the liver in ocular melanoma. So these are the sort of efforts that have really ramped up during COVID-19 and compensated for the de-escalation of wet lab activities. So that's one area that we've really focused on. The second research area that we've focused on is really collaboration. And that's involved communication with other melanoma research groups. And so we've really kicked around ideas and also what data do we have that we are able to share with other research groups, with the idea that if we join forces and combine data, we'll be stronger together. And it might be data, say, for a stronger research publication that we could write at this point in time. Or it might be research data that if we combined, we can submit a grant application. And so we've really spent a lot of time communicating and working with other research groups, just looking for ways in which we can combine efforts.
Yeah, that's fantastic. So even though your physical lab you can't go into on a daily basis because that's closed, you're able to pivot and work on some other projects right now that you could, I'm guessing, work from your house in Philadelphia.
That's right. I mean, we've within my research group, we have individuals that are living in New Jersey, in Philadelphia, or on the Pennsylvania side. And we've really set up multiple meetings, typically thematic meetings, that then focus on areas in which we as a group can work together. But we've also set up research meetings with other groups nationwide, even internationally, and set up collaborations that way. And that's led to many new and exciting collaborations with groups in Miami and with groups in University of San Diego. And I'm now even talking with groups in Australia as to how we can really enhance our research activities and keep them going. The other thing that's been good in this time, and I think often a crisis is the ideal time for innovation. And that's really allowed us to just step back and contemplate the work that we've done and what are the really big questions in the field, and are we addressing them? And so it's been a very good time just to contemplate, to step back and reset ourselves for ramping back up lab activities and coming up with new hypotheses and making sure that we really are addressing the critical questions in the melanoma field.
Wonderful. Let's talk a little bit about ramping back up, because I think, you know, most of us, we're going to go back into our offices, we're going to turn the lights on, and things are going to resume back to their normal state, so to speak. But that's not true for a lab. And as you mentioned, the wet lab has really not been able to keep going. And so you have pivoted to these other areas of research. And part of the reason that we are raising these funds through the gala is to provide a research grant back to Thomas Jefferson University to directly support the melanoma research program there and really help reinvigorate it, restart it, re-energize it. So maybe you could speak a little bit to what is it like restarting a lab? I mean, certainly it's not just turning the lights back on, but there's a lot of work involved. Could you share a little bit about what that's like?
Absolutely. So we're literally going through this now. From late March, the lab activities were de-escalated, and what that means is that we were only able to perform essential activities. And that meant individuals going in typically for 1 to 2 hours twice per week. And it was really checking on those essential stocks that we had. We've just gotten the green light to start to escalate lab activities or wet lab activities back up again. But we obviously have to do that while maintaining appropriate physical social distancing. So a lot of work really went into the planning of that, the setting up of schedules for who's going to go when, the coordination of those schedules. And then also resetting the equipment and making sure that all those equipment are working, they're functional, we're able to get melanoma cells and grow them, and that we have the appropriate resources and reagents to perform the experiments that we need to perform. But as I said, I'm excited about the re-escalation of lab activities, because we've got a whole series of new ideas and hypotheses that we're just poised to test. And so I think the next few months are going to be very, very exciting.
That's wonderful. So a lot of the people who are watching this right now are not scientists. Many are patients and caregivers, and they're probably thinking to themselves, okay, what is exactly a wet lab? Can you give a little more detail as to what exactly a wet lab would be or is?
Certainly. So the wet lab activities are usually focused around bench work. The traditional view is an individual wearing a white lab coat and really pipetting samples in between tubes, but it's a lot more than that. We have equipment and facilities in which we can analyze and grow cells. We've got equipment in which we can take a tumor and look at the different cellular populations and the characteristics and the cell surface markers of those different cell populations. And then we've also got equipment in which we can look at very finite details, the genes that are altered within melanoma cells. So there's a lot of specialized equipment that really goes towards the different types of analysis that we perform.
And how many people do you have working in your lab?
I have a very diverse lab. I have a really good group of people. They get on very well, and we have 15 people working in my group. And as I say, it is diverse in terms of their expertise and also what they work on. And so we have a range of different projects covering the gamut of melanoma research, and then also training at different levels, whether we have individuals that are straight out of college, and we have graduate students that are being trained to get their PhD or their MD PhD. And then also fellows that have gained their PhD and are now doing their postdoctoral fellowship. So we have a range of levels of individuals within the lab.
That's wonderful. So are they all going to come back at once, or are people going to start to slowly come back to the lab? And when do you see the lab being sort of fully operational again in terms of a timeline?
So the way that we've organized it at present is really with social distancing with 1/3 of the lab capacity at one period of time. And So what that means is that we have essentially two shifts within the lab, an early shift and a late shift. And then the individuals that can work remotely are still working remotely. So there's really three different groups: the remote group, and then the two other groups that are going in for fairly short shifts and then doing some remote work. So that's the way that we have it set up.
I bet everyone's excited to go back and start working again, though.
They're very excited to go back and work.
Yeah.
And really because they've had 2 months to really sit down, think about their research and come up with new ideas. Mm-hmm. So I think we're very well poised coming out of this. In terms of when do I think we're going to be fully going again, that is very hard to tell because of the second wave notion, and also I think the difficulties next winter in terms of differentiating COVID symptoms from flu-like symptoms. So we're monitoring all the time, and also the institutional has been great in terms of giving very clear messages and also communicating with other universities in Philadelphia. So there is quite a consensus on how we revamp the work activities.
Absolutely. Safety is always number one, right?
Absolutely.
So I have a couple questions. One, I don't know if you can answer, because a lot of times researchers won't answer this question, but you've mentioned a couple times that you've been really excited to sort of think about new projects and use this time to innovate. Is there anything you can share with us that you're working on, that you're excited about, or that you can't wait to get back into the lab to test without giving anything away?
I'm excited about many things, sure. So yes, I can. And I share ideas because I think you always get feedback on them. So, I mean, one of the ideas that we're really interested in is understanding the population of melanoma cells that tolerate drugs. So melanoma patients that are receiving BRAF inhibitors or BRAF and MEK inhibitors. What are the population of cells that can tolerate that targeted therapy? And I mean, some of this work, some of the characterization of those populations has come out of labs in Philadelphia, but also other labs. We're looking at ways of trying to target those populations, so understanding those populations and then therapeutically targeting them. So we're working very hard on that particular notion with the idea that we can then optimize targeted therapies. So that's one. A second way is, and this second way actually relates a little bit to COVID, and it's ultimately looking to provide a localized inflammation to tumors to improve immunotherapies. So we've heard quite a lot in the last 2 months about the COVID cytokine storm. And this is essentially an overreaction in the whole body of the immune system that can endanger a patient when he or she's fighting an infection. So what we're interested in doing is trying to induce that in a very localized way within the melanoma tumor. So not in the whole body, but just within the tumor. So with the hope that then that will lead to the infiltration of immune cells, and so that the immune cells can really attack that tumor more effectively. So that's an area that we're working on, and really the inflammatory state of melanomas. And then a third area that relates to ocular melanoma, and this is sort of a long-standing interest that is very collaborative with multiple other ocular melanoma groups within the United States, is to understand the role of a gene called BAP1 that's associated with the more aggressive forms of melanoma. And so we're understanding how this gene that's lost in metastasizing ocular melanomas is playing a role in metabolism, but also tumor dormancy. the idea that you get the early dissemination of tumor cells, in the case of ocular melanoma, to the liver, and the notion that those cells might be sleeping, they might be dormant there before they reawaken, and that underlies the delayed clinical presentation of overt metastasis in ocular melanoma patients after treatment of the primary tumor. So those are three areas that we're really excited to work on.
Yeah, those are three amazing projects, and especially the ocular melanoma work. I mean, that's a patient population that desperately needs more research and investment to come up with better treatments for metastatic ocular melanoma.
We work very closely with the medical oncologists at Thomas Jefferson University, and also the Shields at the Wills Eye Institute, and really have developed multiple collaborative team-based approaches that are designed to tackle the research questions within ocular melanoma. And it's, as you say, it's a clinical unmet need in the melanoma field.
Absolutely. Well, thank you. I think that will give a lot of hope to people watching to see these wonderful projects that are still being worked on and that can be really taken to the next level once the lab is open. Can you speak a little bit? We're going to talk a little bit about the gala now, and a little bit about what this award means to you to receive the Humanitarian Award from the Melanoma Research Foundation. And we've changed our model really with funding. Most of the galas, the money we raise then, goes out as a research grant, but it could go to any institution across the United States. And as you know, as a recipient of a MRF research grant before, it's a very competitive process. But in this case, we really want to help restart or help reinvigorate research programs that have been impacted by COVID-19. So that's why the money raised at this event, our virtual gala next week, will go back to TJU in the form of a research grant. But it would be great to hear from you, Dr. Aplin, what this means to you. You've obviously attended the gala before. This will be a different experience, but nevertheless, just as impactful and, in recognizing all of your accomplishments.
Well, first of all, I should really recognize the Melanoma Research Foundation, first of all, for supporting me early on and supporting the efforts in my lab to initiate research in ocular melanoma. My very first ocular melanoma grant came from the Melanoma Research Foundation and CureOM. And so I am forever grateful for that, because I think that has been the launching pad for us to really initiate lots of team-based efforts. With regards to the gala and the award, I mean, this is such an honor. I'm just humbled to receive such an honor. I think it's really recognition for the work that my group has done over the last 2 decades. I think it's recognition of the ability of the individuals within my group to work with each other and also connect with other research groups. And I think that we've done a really amazing job at really trying to look at how we can partner with other melanoma research groups. I think it also reflects on our ability to interact with the clinicians as well. Some of the research from our lab has formed the basis for new clinical trials that have started. And that's always just such a thrill to see when a clinical trial gets approved and starts accruing patients. And then thirdly, I think the ability of the lab to communicate with patients and to communicate with caregivers. I think it's a reflection on the openness of the lab to interact with people, to explain their research, and to give lab tours, and really interact with many different communities at different levels. So it's a real honor. It really is.
Well, we are so happy to honor you, and you've had such a nice long-standing relationship with the MRF over the years, participating on our scientific advisory committee, which you are currently doing, involved with our CureOM initiative, which is focused solely on ocular melanoma, and just being a supporter of all the advocacy work we do. So we are completely thrilled to honor you next week, and we hope that there's a nice big virtual following where you can virtually feel the applause that you'll be receiving. We want to thank you, Dr. Aplin, for spending your time with us today for our first Meet the Researcher. Is there anything else you want to share with our world before we sign off?
Just to say that we are starting to ramp up research activities. We're very excited to do that. I think there will be a tremendous activity in the next 12 months as we really test our new ideas, and we're very excited to do that.
Wonderful. Well, thank you, Dr. Aplin. And as a reminder to everyone watching, the first annual Virtual Philadelphia Gala will be happening next week on June the 4th. We will be honoring Dr. Andrew Aplin, and the funds raised will go to support all the good work you just heard about him and his team at TJU. So I really hope that you all will join us, support the event. We have some wonderful auction items. We have some wonderful trips that you can have a chance to win. And we just want everyone to know that the Melanoma Research Foundation is still standing behind our research community, funding research, as well as our patient community. So thank you all for joining us today, and we hope to see you next week.
Thank you.